Our next talk is by Bob Lansdorp from the UCSB Materials Department. How do nanomotors unzip your genes?
How do nanomotors unzip your genes? In this talk, I'll be focusing on one particular nanomotor whose job it is to turn double-stranded DNA to two single strands. So at the heart of biology is replication, which is the process of converting double-stranded DNA into two identical copies. In this computer model, we can see how nanomotors work together to replicate DNA coming in from the left here and being copied in two identical copies here. Right now, around 1 trillion nanomotors like this one are operating inside of each of you, replicating your DNA. However, if you get infected with a virus, that virus will inject nanomotors very similar to yours into your cells to replicate its DNA. So it's my hope that by understanding exactly how these nanomotors work, we can design drugs to target the viral nanomotors and thereby cure diseases. So in this talk, I'll be focusing on this blue nanomotor at the front here, whose job it is to turn double-stranded DNA coming in into two single strands. And the process of understanding this to generate models like this one involves understanding both the structure and how it moves. So here's a crystal structure that shows the 3D structure of this nanomotor. What you can see is that there's 6 subunits around the outside and some DNA in the center. And so from X-ray crystal structures like this one, we can see the three-dimensional structure in amazing detail. But we're still left wondering, well, how does it move? And the truth is, nobody really knows. And there's models been proposed. For example, here's one model which shows that the 6 subunits of the nanomotor take turns binding to DNA. But is this really how nanomotors move? In our lab, we've designed a new experimental instrument which aims to answer this question. It may look like an ordinary microscope, but actually I've spent the last 2 years customizing it specifically for the purpose of measuring single nanomotors. So with this new instrument, we may wonder, so how does this thing work? Well, what we can do is we can load nanomotors, shown here, onto DNA strands. As these nanomotors start to unzip DNA, we can watch the height of these bead particles move. So this is some experimental data that I collected with this new instrument that actually shows the position of a single nanomotor versus time. So in this data, we can actually start to wonder, well, how does this thing move? Does it move in a straight line with a constant speed, or maybe there's some steps going on? And by performing more experiments like this in the coming years of my PhD, I aim to answer this kind of question and get to the bottom of how do these nanomotors move. With answers to these kinds of questions, we'll expand our understanding of biology and perhaps in the future design new antiviral drugs. In conclusion, with the new experimental tools we've developed here at UCSB, I aim to answer the question, how do nanomotors unzip your genes?